Hello, I am Rakesh, PhD scholar from IIT Bombay. I will be presenting our work on how suitable are clustering methods for functional annotation of protein. The core idea is simple. Can we use unsupervised learning to cluster proteins based on sequence features? Let us start with motivation. Protein sequence databases are increasing rapidly, but the annotations lags behind. Many sequences have no known function. This is where unsupervised learning comes with a hopes to cluster protein sequences such that they reflect molecular functions, leading to transfer of annotation. The objective is to assess multiple clustering algorithms and determine which one are suitable for this task using sequence-derived features, specifically pseudo-amino acid composition, which captures both amino acid composition and sequence order information. To test clustering algorithms, we have chosen a diverse set of 15 datasets. These form 5 groups based on similarities in sequence, structure, and function. We have tested 9 algorithms. These are available in sklearn platform. Only 5 were found to be suitable, shown in green color boxes. The workflow goes like this. First, sequences were downloaded and preprocessed. Then, pseudo-amino acid composition features were extracted, which is a 50-dimensional vector. This acts as an input for clustering algorithm. For each clustering algorithm, the optimum number of clusters were chosen based on the highest Shillard coefficient calculated across different number of clusters ranging from 2 to 10. If the database assigned labels were available, the clustering algorithm with highest FMI was selected as optimal. In case of affinity propagation, only contingency matrix was used to assess the clustering results. Now I'll discuss the results. The clustering quality was evaluated using intrinsic metric to determine the optimum number of clusters. Of 3 intrinsic metrics, Shillard coefficient was chosen to calculate optimum number of clusters k. Since other 2 metrics were unbounded and CH tend to show biased behavior, that is, as number of clusters k increases, CH monotonically decreases, favoring lower values of k regardless of clustering quality. Among Shillard coefficient values for different number of clusters k, we choose k with the highest Shillard coefficient value as optimal, since Shillard coefficient did not reach its upper bound for all the datasets. Now we'll discuss the suitability of mean shift, dbScan, optics, and Birch. The reason for not finding these algorithms suitable for our study, as most or sometimes all the data points are in a single cluster. This may be due to the default parameter not suitable for our datasets or features. For each selected algorithm, we have examined how cluster patterns evolved with increasing value of k. We observed that agglomerative clusters closely mimics the phylogenetic tree, that is, cluster split progressively without recombination. In contrast, other datasets show some phylogenetic-like behavior, but with approximately 10% of the sequence rejoining to form new clusters. Among the clustering algorithms that do not require user-defined key, only affinity propagation was selected, as it was able to segregate proteins in half of the datasets despite producing large number of clusters, as these clusters exhibit homogeneity. Shillard coefficient and FMI index were not suitable for evaluating affinity propagation, as the number of clusters k is determined internally by the algorithm and FMI tends to be lower for large number of clusters. I will discuss the results obtained for selected clustering algorithms. Based on the Shillard coefficient and FMI metric, these clustering algorithms effectively distinguish protein families in group 1, group 2 except proteases and in group 5 except GPCR. It partially separates families in group 3 but failed in group 4 and proteases due to finer function and in GPCR1 due to common transmembrane region. Agglomerative clustering, k-means and Gaussian mixture perform equally well across datasets. The conclusion recommendations are: the clustering algorithm is suitable for annotating both molecular function and glycosylation hydrolysis family. Pseudo-amino acid composition features are effective in distinguishing proteins with similar molecular function. To determine optimum number of clusters k for k-means, Gaussian mixture and agglomerative clustering, choose k with highest Shillard value. To select best performing algorithms among k-means, Gaussian mixture and agglomerative, Use the highest FMA. For the final clustering output, prepare the top performer from the previous conclusion along with affinity propagation with contingency matrix. For more details, you can check bioRK with same titles. Thank you.